T cells or T lymphocytes are a type of lymphocyte that play a central role in cell-mediated immunity. They can be distinguished from other lymphocytes, such as B cells and natural killer cells, by the presence of a T cell receptor on the cell surface. They are called T cells because they mature in the thymus. There are several subsets of T cells, each with a distinct function. Types: helper. T helper cells assist other white blood cells in immunologic processes, including maturation of B cells into plasma cells and memory B cells, and activation of cytotoxic T cells and macrophages. These cells are also known as CD4 plus T cells because they express the CD4 glycoprotein on their surface. Helper T cells become activated when they are presented with peptide antigens by MHC class II molecules, which are expressed on the surface of antigen-presenting cells. Once activated, they divide rapidly and secrete small proteins called cytokines that regulate or assist in the active immune response. These cells can differentiate into one of several subtypes, including Th1, Th2, Th3, Th17, Th9, or TFH, which secrete different cytokines to facilitate a different type of immune response. Signaling from the APC directs T cells into particular subtypes. Cytotoxic T cells destroy virally infected cells and tumor cells, and are also implicated in transplant rejection. These cells are also known as CD8 plus T cells since they express the CD8 glycoprotein at their surface. These cells recognize their targets by binding to antigen associated with MHC class I molecules, which are present on the surface of all nucleated cells. Through IL-10, adenosine and other molecules secreted by regulatory T cells, the CD8 plus cells can be inactivated to an anergic state, which prevents autoimmune diseases. Memory T cells are a subset of antigen-specific T cells that persist long-term after an infection has resolved. They quickly expand to large numbers of effector T cells upon re-exposure to their cognate antigen, thus providing the immune system with memory against past infections. Memory T cells comprise three subtypes: central memory T cells and two types of effector memory T cells. Memory cells may be either CD4 plus or CD8 plus. Memory T cells typically express the cell surface protein CD45 or O. Regulatory T cells, formerly known as suppressor T cells, are crucial for the maintenance of immunological tolerance. Their major role is to shut down T cell-mediated immunity toward the end of an immune reaction and to suppress autoreactive T cells that escaped the process of negative selection in the thymus. Two major classes of CD4 plus Treg cells have been described: FOXP3 plus Treg cells and FOXP3 Treg cells. Regulatory T cells can develop either during normal development in the thymus, and are then known as thymic Treg cells, or can be induced peripherally and are called peripherally derived Tregs. These two subsets were previously called naturally occurring, and adaptive, or induced, respectively. Both subsets require the expression of the transcription factor FOXP3, which can be used to identify the cells. Mutations of the FOXP3 gene can prevent regulatory T cell development, causing the fatal autoimmune disease IPEX. There are several other types of T cell with suppressive activity, but which do not express FOXP3. These include Th1 cells and Th3 cells, which are thought to originate during an immune response and act by producing suppressive molecules. Th1 cells are associated with IL-10, and Th3 cells are associated with TGF-beta. Natural killer T cells bridge the adaptive immune system with the innate immune system. Unlike conventional T cells that recognize peptide antigens presented by major histocompatibility complex molecules, NKT cells recognize glycolipid antigen presented by a molecule called CD1D. Once activated, these cells can perform functions ascribed to both Th and Tc cells. They are also able to recognize and eliminate some tumor cells and cells infected with herpes viruses. Mucosal-associated invariant T cells are a special type of T cell. Gamma delta cells. T cells. Gamma delta T cells represent a small subset of T cells that possess a distinct T cell receptor on their surface. A majority of T cells have a TCR composed of two glycoprotein chains called I+ or minus and I² TCR chains. However, in I³ I T cells, the TCR is made up of one I³ chain and one I chain. This group of T cells is much less common in humans and mice, 2% of total T cells, and are found in the highest abundance in the gut mucosa, within a population of lymphocytes known as intraepithelial lymphocytes. In rabbits, sheep, and chickens the number of gamma delta T cells can be as high as 60% of total T cells. The antigenic molecules that activate I³ I T cells are still widely unknown. However, I³ I T cells are not MHC restricted and seem to be able to recognize whole proteins rather than requiring peptides to be presented by MHC molecules on antigen-presenting cells. Some marine I³ I T cells recognize MHC class IB molecules though. Human Vi³9/Vi2 T cells, which constitute the major I³ I T cell population in peripheral blood, are unique in that they specifically and rapidly respond to a set of non-peptidic phosphorylated isoprenoid precursors, collectively named Phospho Antigens. Phospho Antigens are produced by virtually all living cells. The most common Phospho Antigens from animal and human cells are isopentenyl pyrophosphate and its isomer dimethylallyl pyrophosphate. Many microbes produce the highly active compound hydroxy-DMAPP and corresponding mononucleotide conjugates. In addition to IPP and DMAPP, plant cells produce both types of phospho antigens. Drugs activating human Vi³9/Vi2 T cells comprise synthetic phospho antigens and aminobisphosphonates, which upregulate endogenous IPP-DMAP. Development in the thymus. All T cells originate from hematopoietic stem cells in the bone marrow. Hematopoietic progenitors from hematopoietic stem cells populate the thymus and expand by cell division to generate a large population of immature thymocytes. The earliest thymocytes express neither CD4 nor CD8, and are therefore classed as double-negative cells. As they progress through their development they become double-positive thymocytes, and finally mature to single-positive thymocytes that are then released from the thymus to peripheral tissues. About 98% of thymocytes die during the development processes in the thymus by failing either positive selection or negative selection, whereas the other 2% survive and leave the thymus to become mature immunocompetent T cells. There are increasing evidences that microRNAs, which are small non-coding regulatory RNAs, could impact the clonal selection process during thymic development. For example, it was found that miR-181 plays a role in the positive selection of T lymphocytes. The thymus contributes fewer cells as a person ages. As the thymus shrinks by about 3% a year throughout middle age, there is a corresponding fall in the thymic production of naive T cells, leaving peripheral T cell expansion to play a greater role in protecting older subjects. Beta selection, common lymphoid precursor cells that arrive at the thymus and become known as T cell precursors. When they begin to express c-KIT and CD44, they become known as DN1 thymocytes. At this stage they must create a unique T cell receptor through V/D/J recombination. As the TCRI² locus begins to be rearranged, the DN1 cell begins to express CD25 and becomes a DN2 thymocyte. At this point a T cell will progress toward either the helper killer lineage or the I³ I T cell lineage. The DN2 cell begins to express CD3 and stops expressing c-KIT and CD44, becoming a DN3 thymocyte. At this stage the thymocyte must produce a TCRI² beta chain that can be translated into protein and travel to the cell surface with a pre-T alpha chain. If this occurs, the cell has passed beta selection and will stop expressing CD25. It will also rapidly proliferate. The cell will start to express both CD4 and CD8 and becomes known as a DP thymocyte. Positive selection selects for T cells capable of interacting with MHC. Double-positive thymocytes move deep into the thymic cortex, where they are presented with self-antigens. These self-antigens are expressed by thymic cortical epithelial cells that express activation-induced deaminase on both MHC molecules on the surface of cortical epithelial cells. Only those thymocytes that interact with MHCI or MHCII appropriately will receive a vital survival signal. All that cannot will die by death by neglect. This process ensures that the selected T cells will have an MHC affinity that can serve useful functions in the body. The vast majority of all thymocytes end up dying during this process. A thymocyte's fate is determined during positive selection. Double positive cells that interact well with MHC class II molecules will eventually become CD4+ cells, whereas thymocytes that interact well with MHC class I molecules mature into CD8+ cells. A T cell becomes a CD4+ cell by downregulating expression of its CD8 cell surface receptors. If the cell does not lose its signal through the ITAM pathway, it will continue downregulating CD8 and become a CD4+, single positive cell. But, if there is a signal interruption, the cell stops downregulating CD8 and switches over to downregulating CD4 molecules instead, eventually becoming a CD8+, single positive cell. This process does not remove thymocytes that may cause autoimmunity. The potentially autoimmune cells are removed by the process of negative selection, which occurs in the thymic medulla. Negative selection removes thymocytes that are capable of strongly binding with self-MHC peptides. Thymocytes that survive positive selection migrate towards the boundary of the cortex and medulla in the thymus. While in the medulla, they are again presented with a self-antigen presented on the MHC complex of medullary thymic epithelial cells. MTECs must be Air+ to properly express self-antigens from all tissues of the body on their MHC class I peptides. Some MTECs are phagocytosed by thymic dendritic cells. This allows for presentation of self-antigens on MHC class II molecules. Thymocytes that interact too strongly with the self-antigen receive an apoptotic signal that leads to cell death. However, some of these cells are selected to become Treg cells. The remaining cells exit the thymus as mature naive T cells. This process is an important component of central tolerance and serves to prevent the formation of self-reactive T cells that are capable of inducing autoimmune diseases in the host. In summary, I² selection is the first checkpoint, where the T cells that are able to form a functional pre-T cell with an invariant alpha chain and a functional beta chain are allowed to continue development in the thymus. Next, positive selection checks that T cells have successfully rearranged their TCRI+± locus and are capable of recognizing peptide MHC complexes with appropriate affinity. Negative selection in the medulla then obliterates T cells that bind too strongly to self-antigens expressed on MHC molecules. These selection processes allow for tolerance of self by the immune system. Typical T cells that leave the thymus are self-restricted, self-tolerant, and singly positive. Activation of CD4+ T cells occurs through the simultaneous engagement of the T cell receptor and a costimulatory molecule on the T cell by the major histocompatibility complex peptide and costimulatory molecules on the APC. Both are required for production of an effective immune response. In the absence of co-stimulation, T cell receptor signaling alone results in anergy. The signaling pathways downstream from costimulatory molecules usually engages the PI3K pathway generating PIP3 at the plasma membrane and recruiting pH domain containing signaling molecules like PDK1 that are essential for the activation of PKCTH-ETA, and eventual IL-2 production. The first signal is provided by binding of the T cell receptor its cognate peptide presented on MHCII on an antigen-presenting cell. MHCII is restricted to so-called professional antigen-presenting cells, like dendritic cells, B cells, and macrophages, to name a few. The peptides presented to CD8+ T cells by MHC class I molecules are 8 to 9 amino acids in length. The peptides presented to CD4+ cells by MHC class II molecules are longer, usually 12-25 amino acids in length, as the ends of the binding cleft of the MHC class II molecule are open. The second signal comes from co-stimulation, in which surface receptors on the APC are induced by a relatively small number of stimuli, usually products of pathogens, but sometimes breakdown products of cells, such as necrotic bodies or heat shock proteins. The only co-stimulatory receptor expressed constitutively by Norvay T cells is CD28, so co-stimulation for these cells comes from the CD80 and CD86 proteins, which together constitute the B7 protein, on the APC. Other receptors are expressed upon activation of the T cell, such as OX40 and ICOS, but these largely depend upon CD28 for their expression. The second signal licenses the T cell to respond to an antigen. Without it, the T cell becomes anergic, and it becomes more difficult for it to activate in future. This mechanism prevents inappropriate responses to self, as self-peptides will not usually be presented with suitable co-stimulation. The T cell receptor exists as a complex of several proteins. The actual T cell receptor is composed of two separate peptide chains, which are produced from the independent T cell receptor alpha and beta genes. The other proteins in the complex are the CD3 proteins, CD3I micron I cubed and CD3I micron I heterodimers and, most important, a CD3I paragraph homodimer, which has a total of 6 ITAM motifs. The ITAM motifs on the CD3I paragraph can be phosphorylated by LCK and in turn recruit ZAP70. LCK and or ZAP70 can also phosphorylate the tyrosines on many other molecules, not least CD28, LAT and SLP76, which allows the aggregation of signaling complexes around these proteins. Phosphorylated LAT recruits SLP76 to the membrane, where it can then bring in PLCI cubed, VAV1, ITK and potentially PI3K. PLCI cubed cleaves PI, 4, 5, P2 on the inner leaflet of the membrane to create the active intermediaries diacylglycerol, inositol 1, 4, 5 trisphosphate. PI3K also acts on PIP2, phosphorylating it to produce phosphatedly inositol 3, 4, 5 trisphosphate. DAG binds and activates some PKCs. Most important in T cells is PKCI, critical for activating the transcription factors NFIB and APRIL1. IP3 is released from the membrane by PLCI cubed and diffuses rapidly to activate calcium channel receptors on the ER, which induces the release of calcium into the cytosol. Low calcium in the endoplasmic reticulum causes STIM1 clustering on the ER membrane and leads to activation of cell membrane CRAC channels that allows additional calcium to flow into the cytosol from the extracellular space. This aggregated cytosolic calcium binds calmodulin, which can then activate calcineurin. Calcineurin, in turn, activates NFAT, which then translocates to the nucleus. NFAT is a transcription factor that activates the transcription of a pleiotropic set of genes, most notable, IL-2, a cytokine that promotes long-term proliferation of activated T cells. PLCI cubed can also initiate the NFIB pathway. DAG activates PKCI, which then phosphorylates CARMA1, causing it to unfold and function as a scaffold. The cytosolic domains bind an adapter CBL10 via CAR domains. That then binds TRAF6, which is ubiquitinated at K63 template, RP513/523. This form of ubiquitination does not lead to degradation of target proteins. Rather, it serves to recruit an EMO, IKI plus or minus and I2, and TAB1-2/TAK1. TAK1 phosphorylates IKKI2, which then phosphorylates EB, allowing for K48 ubiquitination, leads to proteasomal degradation. RAL-A and P50 can then enter the nucleus and bind the NFIB response element. This coupled with NFAT signaling allows for complete activation of the IL-2 gene. While in most cases activation is dependent on TCR recognition of antigen, alternative pathways for activation have been described. For example, cytotoxic T cells have been shown to become activated when targeted by other CD8 T cells, leading to tolerization of the latter. In spring 2014, the T cell activation in space experiment was launched to the International Space Station on the SpaceX CRS-3 mission to study how deficiencies in the human immune system are affected by a microgravity environment. Disorders, deficiency. Causes of T cell deficiency include lymphocytopenia of T cells and/or defects on function of individual T cells. Complete insufficiency of T cell function can result from hereditary conditions such as severe combined immunodeficiency, Omenn syndrome, and cartilage hair hypoplasia. Causes of partial insufficiencies of T cell function include acquired immune deficiency syndrome, and hereditary conditions such as DiGeorge syndrome, chromosomal breakage syndromes, and B-cell and T-cell combined disorders such as ataxia telangiectasia and Wiskott-Aldrich syndrome. The main pathogens of concern in T-cell deficiencies are intracellular pathogens, including herpes simplex virus, mycobacterium and listeria. Also, fungal infections are also more common and severe in T-cell deficiencies. Cancer. Cancer of T cells is termed T cell lymphoma, and accounts for perhaps 1 in 10 cases of non-Hodgkin lymphoma. The main forms of T cell lymphoma are extranodal T cell lymphoma, cutaneous T cell lymphomas, Czar-Copyright-Zary syndrome and mycosis fungoides, anaplastic large cell lymphoma, angioimmunoblastic T cell lymphoma. Additional images. See also, gut-specific homing, immunoblast, references. External links, Immunobiology, 5th edition, NIAID.NI.gov. The immune system, T cell group. Cardiff University, successful. Treatment of metastatic melanoma with autologous CD4 plus T cells against NYESO1. The Center for Modeling Immunity to Enteric Pathogens, Davies A.J. The Tale of T Cells. Immunology Today 14, 137 139 doi, 10.1016/0167-5699, 93, 90216-8. PMIDA 8466629A.